So I just thought I'd take a little bit of time to show you what Sean and I are doing in the lab these days in terms of molecular biology. And the challenge we have is that we want to know what fungus is colonizing these roots. And just by looking at the root or staining it and looking inside won't tell us the identity of the fungus. So for that, we're required to use molecular techniques, and we're using something called PCR for that. It stands for polymerase chain reaction, and essentially allows us to go from this to this. And this is a chromatogram of an arbusculum mycorrhizal fungus that we recently sequenced. And it shows us the quality of the sequence here and the actual sequence. So these are the building blocks of the nucleotides that we all have and that makes us who we are, really. And PCR was really invented by a man in California called Kary Mullis in the early 80s. And it has revolutionized biology in that we can actually go out in the field and look at sequences, or actually get sequences from environmental samples where the amount of DNA is so low. It's a technique to exponentially increase the DNA concentration, which allows us to sequence and find out who's there. So essentially the first step to getting a sequence of fungi colonizing these roots is to extract the DNA from this root. And we use different methods depending on what we're interested in, but the very simplest method is to essentially just cut up small root fragments and crush the roots and try to remove proteins and other things that could inhibit the PCR. And in the end, we have a mixture of plant and fungal DNA, or any other DNA that might be in this root. Now, if we amplified all the DNA and send it off to sequencing, it would just be a mixture of plant and fungal DNA. So we need to make sure that we're only amplifying fungal DNA. And we can do that by designing specific primers in the PCR reaction. And essentially what the PCR reaction is that what it allows us to do is exponentially increase the target DNA that we're interested in. So obviously we have a mixture of double-stranded DNA, plants, fungi, bacteria, but we're interested in only a specific group. So the PCR reaction essentially has 3 different steps. The first step, and it all happens in the thermocycler that Sean is going to show you later on. The first step is that we heat the double-stranded DNA to 95 degrees, and what that does is to separate it into single-stranded DNA. By lowering the temperature to 55 degrees, it allows our specific primers to bind, and we know the primer has been designed to bind to specific regions in this fungal DNA. So at 55 degrees, it binds to the single-stranded DNA, and then we have that's called the annealing step, and then we have the elongation step, and that's usually at 72 degrees Celsius. And what that does is that the primer is bound, and then we have an enzyme called TAC, which was isolated from geothermal areas in Yellowstone. And the good thing with TAC is that it can tolerate and operate under higher temperatures. So and what TAC does is that it reads the DNA sequence here. It might be G, A, C, T. It is able to read that sequence and add the nucleotides, the building blocks that we supply in the PCR onto that. And in the end, after the elongation step, we have again a double-stranded DNA. And we go through a second cycle. It goes up to 95 degrees. We have a separation into single-stranded DNA. 55 degrees, the primers bind again, and the elongation step. And there we have, again, double-stranded DNA as a result. And by running about 30 cycles or so of this, in the end, we have enough DNA that we can see on a gel. And Sean is going to show you some gel pictures that we've generated in the lab as well. And that is something we can send off to sequencing, and we get the sequence of the fungus that is colonizing this small root piece, and we can get an idea of the fungal community we have out at MPG Ranch.
All right, well, Ilva gave you a little bit of a background on her project, the idea for this project, so I'll talk a little bit more about the practical day-to-day in the lab kind of stuff. So, as we talked about before, we're using polymerase chain reaction to look at these organisms. They're obviously microorganisms. You can't just go outside and do a survey and say there's a cheatgrass and there's a knapweed and you can count them. You have to look at them, look at their molecules to be able to quantify who's there and what abundance they're there. So we get the sequences and we're able to send those off and get them back and look at the community composition of these microbes. So the way we do that, we're doing next generation sequencing called 454 sequencing. And it's a very sensitive method where you can get hundreds of thousands of sequences back. And you want to be very careful when you send them samples because, as we talked about before, Taq polymerase was isolated from Yellowstone out of this hot thermal pool, Thermus aquaticus, or T. aquaticus, or Taq. That's how it gets its name. It's able to operate at high temperatures. It's perfect for the PCR reaction because you're cycling between really hot and medium range temperatures. But the problem with normal Taq is it's got an error rate of about 2 errors per 100,000 base pairs. So when it's writing the genetic code, every 100,000 base pairs or so, it makes an error. And that won't do for sending off and getting back hundreds of thousands of sequences or millions of base pairs, because you may have a change in a certain sequence, but it may just be due to the error of the enzyme and not actually reflect the sequence itself. So you may call this a different species just because the Taq made a mistake. So we bought a high fidelity Taq polymerase, or a proofreading Taq, which is able to run along the DNA and look back at errors it's made, chew up what it's done, and then redo it. So it's much more trustworthy. It makes only 4 errors in 10 million base pairs, about 2 orders of magnitude better. And that'll give us better data in the end. So this is the machine that allows us to do PCR. It's the thermocycler. It opens up and you throw your tubes inside, and it's got a metal plate that can heat and cool very rapidly. And you can program it, it's a computer essentially, and you program it to run cycles of temperatures. So you want to do 95 degrees for 30 seconds, 58 degrees for a minute, and then 72 degrees for 50 seconds. And you do 30 cycles of that. And you get an exponential increase in the amount of DNA that you're targeting in your PCR reaction. So you go from 1 sequence in your little tube to a billion sequences of that same sequence. And you need that high copy number in order to send it out for the sequencing to get that chromatogram you saw before. You can't just take 1 sequence, give it to them, and say, give me a sequence. It just doesn't work that way. You have to have a lot to be able to see it. This is our gel rig. So once we've run our PCR reaction, we need to be able to see whether or not we got our desired product. And we're going to know how big it's going to be. We know where our primers stick to the template DNA or the genomic DNA, and we know the area between those primers is a certain length, say 900 base pairs. So we create this gel by melting agarose into water. It's essentially like making Jell-O. And when it's still molten or liquid, you add this dye, which is called ethidium bromide. And then you pour the gel into this gel rig and then put a comb in, which is this little thing here, which puts little indentations into your gel. And then you let it solidify, and it's ready to run. And the ethidium bromide stain that we add to the gel is what we call an intercalating dye. It has this ring structure. It's an arm that kind of jams into the double-stranded DNA. And it jams in at all the little junctions, and when it's excited by ultraviolet light, it fluoresces, or it gives off a red light that we can see with our eyes. So we can really visualize the DNA using that dye, and it's very sensitive. So once our gel's made and the wells are in, we can pull out our comb, add a buffer solution. It's just a salt solution. And then we add our DNA samples to each well, along with a molecular ladder, which you just buy at a molecular biology company. And it's it's a pre-made mixture of DNA fragments of known sizes. Say 1,000 base pairs, 500 base pairs, 300 base pairs, 100 base pairs, and so on. And so it's a ladder that runs next to your samples, and you can match your sample to the ladder to see what is the exact size of the sample that you're running. So if you are looking for a 900 base pair sample and you look at your ladder, you can tell, yes, that's what I want. That is the exact fragment. So once your gel is loaded and in the rig, and you have your solution, your salt solution in the rig, you plug in to your power source. Negative here and positive here. Your DNA samples are on the negative pole, and DNA is itself negatively charged, and it's pulled towards the positive end of your rig. Opposites attract. So the positive charge will actually pull your DNA fragments through this jello, and the larger your DNA is, the more slowly it moves through this jello. And the small fragments will move a lot more quickly, so you can separate out based on size your different DNA fragments. So once we have our gel that's been run at a certain voltage for a certain amount of time and your DNA fragments are separated, then you want to be able to visualize them. So we have this little gel dock here. We kind of jerry-rigged up a cardboard box to keep light out from the outside when we're taking pictures of our gels. So we open up our box, and inside we have this platform, and the platform has a glass plate, and underneath that glass plate is a UV lamp. So when you turn it on, UV light streams upward, and there's a plastic covering that kind of folds over the top of the glass plate that protects us, because UV obviously damages our skin and our eyes, and it is blocked from going through this plastic plate. So we open up the plastic plate, we place our gel on top of the glass surface, close it up, and then adjust our camera and switch on our UV light source. And then once that's done, we just focus the image on the camera with the computer and take our picture, and we have our gel picture stored on the computer. So this is a picture of one of the gels from the 454 sequencing experiment. As you can see, here we have negative wells, so these samples didn't yield a fragment, whereas these bright bands are our fragment of interest. On the far left is the DNA ladder, which tells us the size of our fragments, and the fragment of interest is 900 base pairs, which aligns nicely to the 1,000 base pair band, or closely to the 1,000 base pair band of the ladder. So we know that this is the fragment we want, and the fragment is a part of the large subunit of ribosomal DNA in avascular mycorrhizal fungi. And that particular sequence is very good at distinguishing species. So we'll be able to distinguish between species using these sequences when we send them off for pyrosequencing or 454 sequencing. So in the end, we will hopefully have one of these bands for every single one of the sites that Ilva sampled. We'll optimize these so that we get bands, and once we have AMF DNA from every single site, we'll then pool them all together into a single tube. So it's all together in one tube, and each one of these bands is going, from each site, is going to have a separate tag on it. It's been labeled like a barcode using DNA. So we can distinguish the sequences from site 1 with that tag, and site 2, site 3, site 4, and there are 23 sites. So all 23 are labeled. That way we can pool them all into one tube, send that off to the sequencing facility, and they run it all on one reaction, and we get hundreds of thousands of sequences back. Using a computer, we sort those sequences into bins for each site, and then we can have a species list for every single site and then analyze that data.